Welcome to today's scientific lecture on a fascinating phenomenon that occurs in your kitchen every day, the salt-acid amplification effect. Why does squeezing lemon juice on your food make it taste saltier? Have you ever noticed that when you add lemon juice to a dish that already contains salt, the food suddenly tastes much saltier than before? This isn't just your imagination. There's real chemistry happening here. Today we're going to dive deep into the molecular interactions between acids and salt that create this remarkable amplification effect. Let's start by asking a fundamental question. What exactly is happening at the molecular level when we combine salt and acid? First, let's talk about salt. Table salt is sodium chloride, NaCl. When dissolved in water or in the moisture on your tongue, it dissociates into positive sodium ions and negative chloride ions. Here we see sodium chloride dissociating. The smaller red sphere represents the sodium cation, and the larger blue-green sphere represents the chloride anion. Sodium chloride dissociates into sodium plus and chloride minus. Now let's talk about acids. Lemon juice contains citric acid, which is a weak organic acid. Acids are proton donors. They release hydrogen ions into solution. Citric acid partially dissociates to release hydrogen ions, also called protons, and citrate anions. This is an equilibrium reaction. The concentration of these hydrogen ions determines the pH of a solution. Lemon juice has a pH of about 2 to 3, making it quite acidic. Now let's explore the first mechanism behind the salt-acid amplification effect. Taste receptor competition on your tongue. Your tongue contains specialized taste receptor cells. There are different types for sweet, sour, salty, bitter, and umami tastes. This simplified diagram shows a cross-section of taste receptor cells. Different receptors respond to different chemical stimuli. Salty taste is primarily detected by epithelial sodium channels, abbreviated ENaC. These channels allow sodium ions to pass through the cell membrane. Here's the key question. How does the presence of acid affect these sodium channels? Recent research shows that hydrogen ions from acids can modulate the activity of these sodium channels. The acidic environment increases the open probability of ENaC channels. The channel protein is shown here in brown. Sodium ions in red attempt to pass through. When hydrogen ions in yellow are present, they interact with the channel and increase its permeability to sodium. The result is that more sodium ions can enter the taste receptor cells, generating a stronger electrical signal that your brain interprets as saltier taste. The second mechanism involves the concept of ionic strength and ion activity. This gets into some fascinating physical chemistry. Let me ask you, what is ionic strength? Ionic strength is a measure of the total concentration of ions in a solution. Ionic strength I equals 1/2 times the sum over all ions of concentration, C sub I, times the charge, Z sub I squared. When you add lemon juice to food, you're not just adding acid, you're adding hydrogen ions and citrate ions, which increases the total ionic strength of the solution. Now here's where it gets interesting. There's a difference between ion concentration and ion activity. Activity is the effective concentration that participates in reactions. Activity A sub I equals the activity coefficient gamma sub I times the concentration C sub I. In very dilute solutions, the activity coefficient is close to 1, meaning activity equals concentration. But as ionic strength increases, this coefficient decreases. According to the Debye-Huckel limiting law, log of gamma sub I equals negative 0.5, 1 times Z sub I, squared times the square root of ionic strength I. This creates an interesting paradox. Higher ionic strength means lower activity coefficients, which would suggest less effective concentration. So why does food taste saltier? The answer lies in competitive binding and displacement. The additional ions from the acid compete for water molecules and change the local environment around sodium ions. The third mechanism involves how pH affects the conformation of proteins in food, which changes how salt interacts with your taste receptors. Food proteins contain amino acid residues with charged groups. These include carboxyl groups and amino groups that can be protonated or deprotonated depending on pH. At higher pH on the left, carboxyl groups are deprotonated as COO-. When we add hydrogen ions and lower the pH, These groups become protonated as COOH. When you add acid and protonate these groups, you reduce the negative charge on the protein surface. This reduces electrostatic repulsion between protein molecules. The result is that proteins can unfold slightly and expose more binding sites. This changes how sodium ions interact with the food matrix and with your taste receptors. Think of it like this. Imagine a clenched fist representing a tightly folded protein. Adding acid is like relaxing the fingers, exposing the palm. Now there's more surface area for sodium ions to interact with. The fourth mechanism involves your saliva and the temporal dynamics of taste perception. This is often overlooked but crucial to understanding the full effect. Your saliva is slightly alkaline, typically with a pH between 6.5 and 7.5. It contains buffers like bicarbonate and phosphate ions. Bicarbonate ion plus hydrogen ion forms carbonic acid in an equilibrium reaction. When you add lemon juice to food, the acid temporarily overwhelms your saliva's buffering capacity. The local pH on your tongue drops significantly. This creates what we call a temporal pH gradient. The pH changes over time as your saliva works to neutralize the acid. This curve shows salt taste intensity over time. There's a peak in the middle representing optimal amplification. Below this, the effect is weak. Above this, acid taste dominates and masks the salt. Research shows that peak salt intensity occurs approximately 2 to 5 seconds after the food contacts your tongue, right when the pH is at its lowest. Now let's look at some quantitative analysis. Can we actually measure and predict this amplification effect? Sensory studies using trained taste panels show that adding citric acid to a sodium chloride solution can increase perceived saltiness by 20 to 40%, depending on concentrations. The change in saltiness intensity can be modeled empirically as delta S equals K times the hydrogen ion concentration to the 0.3 power times the sodium ion concentration, where K is an empirical constant. The exponent of 0.3 indicates that the effect has a sublinear dependence on acidity. Doubling the acid concentration doesn't double the amplification. Let's work through a concrete example. Imagine you have a piece of fish that's been lightly salted with 0.5% sodium chloride by mass. 0.5% sodium chloride by mass corresponds to approximately 85 millimolar concentration. Now you squeeze fresh lemon juice on it. Lemon juice contains about 5% citric acid by mass. At pH 2.5, the hydrogen ion concentration is approximately 10 to the negative 2.5 molar, which is 3.2 millimolar. Using psychophysical models, we can calculate the amplification factor. The amplification factor A equals 1 plus 0.32 times 3.2 to the 0.3 power, which equals approximately 1.38. This means the perceived saltiness increases by about 38% compared to the fish without lemon juice. This scientific understanding has profound practical applications in cooking and food science. One major application is sodium reduction. By strategically using acids, chefs and food manufacturers can reduce sodium content by up to 30% while maintaining the same perceived saltiness. Commercial soup manufacturers now routinely add small amounts of citric acid or vinegar to reduce sodium levels without compromising taste. The second application is flavor layering. The salt-acid amplification doesn't just make food saltier, it creates a more complex, dynamic flavor profile. Many traditional cuisines have discovered this effect through centuries of experimentation. Mediterranean, Southeast Asian, and Latin American cooking all feature acid-salt combinations. Here are 3 classic examples from world cuisine. Ceviche combines lime and salt. Vietnamese food pairs fish sauce with lime. Greek salads feature feta cheese with lemon. All exploit the salt-acid amplification effect. An important question: does this effect work with other acids, or is it specific to citric acid from lemons? The answer is that the effect works with most acids, though the strength varies depending on the specific acid used. Acetic acid from vinegar produces strong amplification, similar to citric acid. This is why vinegar is so commonly used in cooking. Lactic acid from fermented foods like yogurt or sauerkraut produces moderate amplification. It also adds complexity through other flavor compounds. Phosphoric acid found in cola drinks produces weaker amplification. This is partly why cola can taste very sweet despite containing significant sodium. The relative amplification strength goes acetic greater than citric greater than lactic greater than phosphoric. Why the difference? The molecular structure of the acid matters. Smaller, more mobile molecules like acetic acid can interact more readily with taste receptors. Let's examine how concentration affects this phenomenon. Is more acid always better? The answer is no. There's an optimal concentration range for acid. Too little acid produces minimal amplification. Too much acid overwhelms the salt taste entirely. This curve shows salt perception versus acid concentration. There's a peak in the middle representing optimal amplification. Below this, the effect is weak. Above this, acid taste dominates and masks the salt. For most foods, the optimal range is pH 3 to 4. This provides strong amplification without overwhelming sourness. An important consideration is that not everyone experiences this amplification effect equally. There's significant individual variation. Taste receptor genes show genetic polymorphism. They vary between individuals. Some people have more sensitive ENaC channels than others. Super tasters, people with higher density of taste buds and more sensitive receptors, tend to experience stronger salt-acid amplification than average tasters. Age also affects sensitivity. Children generally have more sensitive taste receptors and may experience stronger amplification. Elderly individuals often have reduced sensitivity. This phenomenon has important implications for public health and nutrition policy. High sodium intake is strongly linked to hypertension and cardiovascular disease. The average American consumes 3,400 milligrams of sodium per day, well above the recommended 2,300 milligrams. Strategic use of the salt-acid amplification effect in processed foods could help reduce sodium intake by 25 to 30% without sacrificing palatability. This represents a major public health opportunity. Even a 10% reduction in population sodium intake could prevent hundreds of thousands of cardiovascular events annually. Let me share some exciting current research in this area. Food scientists are developing novel delivery systems that release acid and salt sequentially in the mouth to maximize the amplification effect. Microencapsulation technology allows acids to be released at specific times during chewing, creating dynamic flavor profiles. Neuroscience studies using functional MRI are mapping exactly how the brain processes the combined salt-acid signal, revealing surprising complexity in taste integration. Computational models combining molecular dynamics and psychophysical data can now predict optimal acid-salt combinations for specific foods. Before we conclude, let me address some common misconceptions about this effect. Misconception 1. Some people think this effect is purely psychological or imaginary. As we've seen, there are multiple molecular mechanisms at work. This is real chemistry and physiology. Misconception 2. All acids work equally well. We've shown that different acids produce different levels of amplification based on their molecular properties. Misconception 3. More acid always means more saltiness. There's an optimal concentration beyond which acid taste dominates and actually reduces salt perception. Let's summarize the key scientific principles we've covered today. First, salt-acid amplification is a real, measurable phenomenon with a 20 to 40% increase in perceived saltiness. Second, there are 4 main mechanisms: taste receptor modulation, changes in ionic strength and activity, protein conformational changes, and temporal dynamics with saliva. Third, the effect varies depending on the specific acid used and its concentration, with an optimal range around pH 3 to 4. Fourth, this has important practical applications in cooking, food manufacturing, and public health strategies to reduce sodium intake. Fifth, there's significant individual variation in how people experience this effect due to genetic differences and age-related changes in taste sensitivity. To conclude, the salt-acid amplification effect is a beautiful example of how chemistry, biology, and sensory perception interact in complex ways. What chefs have known intuitively for centuries, that lemon makes food taste saltier, now has a solid scientific foundation spanning multiple disciplines. Understanding these mechanisms allows us to manipulate flavor with unprecedented precision, opening new possibilities in cuisine and nutrition. Thank you for joining me on this deep dive into the science of why squeezing lemon makes food saltier. I hope you'll think about these molecular interactions the next time you reach for that lemon wedge.